Welcome student in another lecture of this week. Now we will in this lecture also we will try to understand some more anomalous structures and how they are formed. So first of all, in this we will start about formation of included phloem. Okay, included phloem means the phloem which is present in the xylem ring, the secondary xylem ring. Okay, like this cambium is forming secondary xylem. Okay, and it is forming secondary phloem on the outer side. So if some patch of secondary phloem is also found in this xylem, it is also known as included phloem. Now, there are actually two methods by which this included formation takes place, by arc cambium formation and by change in the activity of cambium. So, we will discuss both the methods. First one is formation of this included phloem by arc cambium method. See, actually this included phloem is also known as, you should remember that it is also known as interxylary phloem. Okay, so this is known as included phloem. So both the terms might come and it could be asked in any way. So it is known as interxylary phloem, although you have one intraxylary phloem also, that means outside xylem. So that also we will discuss when we will come to that example. So first of all, we will try to discuss about the included phloem by arc cambium method. Some examples are Acaranthus, Chenopodium, Bougainvillea. See, now these examples we have already studied before, and many other anomalous structures were present in these plants also, like in Acaranthus, Bougainvillea. We have studied about medullary bundles, we have studied out about formation of accessory cambia. Now again, one more anomalous structure which is found in these stem is presence of included phloem. Now how this included phloem is developing? See, in this, suppose in Acaranthus and Bougainvillea, what we have studied that there is a formation of accessory cambium. Okay, suppose in Acaranthus, okay, you have two vascular bundles here and one accessory cambia formation was taking place. Okay, and this accessory cambia was cutting xylem on the lower side and phloem on the outer side. Okay, and it was giving a sclerenchyma on the and parenchyma. This one was sclerenchyma, more compact, we can draw it here, and then parenchyma. Okay. Now, when this accessory cambia is developing, okay, sometimes after giving rise to those tissue, at certain points of time, the cambia just beneath this phloem region, okay, in this vascular region, it becomes inactive. Okay, it's not that every cambia will become inactive at one point of time. Any one of them may become inactive. Okay, we can draw a bigger diagram here. Suppose this cambia, which is cutting xylem here, okay, lot of xylem here, and suppose this is, it will become inactive, okay, and then conjunctive tissues are there. Now, if this is becoming inactive, what will happen? Above phloem, now this is inactive, okay. Above this phloem region, okay, another cambium will arise in the arc. Okay, above this phloem definitely parenchymatous tissues are there. Okay, parenchyma is there. Now just adjacent to this phloem, this parenchyma will become meristematic in an arc like fashion. Okay, just forming an arc over phloem. Okay, so we can say that this is arc cambium, and this arc will now join the cambium, previous cambium. Okay, so this will join in the form of arc shape to the cambium which was cutting tissues. Okay, now this cambium, now it will behave as a continuous layer, and it will cut xylem on the lower side and phloem on the outer side. You can see here. So in this way, what will happen if it is if it is cutting tissues like this, this phloem will become embedded, and below this also xylem will be definitely there. Okay, as it is there below this phloem. So below this phloem also xylem will be there, but now this cambia will cut xylem on this side. Okay, and it is normally cutting xylem. So what will happen in this way, this phloem will now become included in the xylem, and now this cambia will behave as normally by cutting xylem and phloem tissue. Now again some more cambium patches can behave in the same way, where the cambia just below phloem will become inactive. Now suppose it can happen here also. Many a times 2-3 cambia can also become inactive at a point. So when this ring is increasing, suppose this ring is all increasing, you will find secondary phloem. Okay, secondary phloem or included phloem embedded in the secondary xylem. So this is the way how this included phloem is produced. Now in bougainvillea, see, as we have discussed before, this case you can see here xylem, see, you can see lot of medullary bundles in bougainvillea. Also we know that lot of medullary bundles are there, and we have discussed before also how different rings of vascular bundles were formed. And in the outer ring, however, the outer ring in which the cambia was forming a secondary layer in which there was sclerenchymatous patches. So in that outer ring, you can see that the included phloem formation has occurred. In Achiranthus, okay, there was immediately a formation of accessory cambia, and it was giving rise to sclerenchyma and then parenchyma on the outer side. So in that case also included phloem formation was occurred. Similarly, in Achiranthus also we have discussed before that in this patch you will see included phloem, small patches of these phloems will be there. Okay, you can see here if we change the color and make it as such. Okay, so this patches of phloem you will see, it will contain the elements which are there in phloem like sieve tubes, companion cells, parenchyma, fibers. So all those things could be there. Then you will have xylem. Okay, vessels are there, and this is the sclerenchymatous layer. Okay, sclerenchyma, which is the conjunctive tissue. Okay, in the medullary bundles you can easily see xylem is on the lower side and phloem is on the outer side. So this is. Now coming to another way of cambium formation. See, in this case the included phloem occurs by change in cambial activity. Okay, there is no arc cambium formation. Okay, that means there is no inactivation of cambium in any region. However, the cambium activity gets changed at certain point of time. So in this case, what happens? Suppose this is the cambia. Okay, and it is normally cutting out xylem. See, now here in this case you can see that there is no accessory cambium formation. Okay, in this case we are talking about the normal cambium. So now this is the cambium. So just remember that now there is no accessory cambium. Now in this case we are not talking about any accessory cambium. Okay, before that all those examples in which accessory cambium was behaving as such, because in those examples there was no normal cambium. But in these plants normal cambium is there. Okay, normal vascular bundles are there. Okay, they are joining their interfascicular and intrafascicular cambium together, and they were giving rise to xylem on the lower side and phloem on the outer side. So here also same thing, but at certain points this cambium will start behaving abnormally. Okay, it won't cease its function. It will start producing phloem instead of xylem. So at this patch some sort of phloem will be produced. So at this point you can say that this cambium will give rise to phloem on both the sides. So now this, and then again after some time this will resume back its activity. So this phloem will now, when more xylem will be produced, this will move on to upper side, and then it will appear that this phloem is actually embedded in the xylem. So this is the way by which included phloem will be formed. Okay. So in this Leptadenia and Salvadora, this type of included phloem formation takes place. Now in Leptadenia, just try to understand it with the diagram. So definitely an outer epidermis will be there. Okay. Then you will have cortex, and then endodermis will be there. And then you will have pericycle, in which we can see the sclerenchymatous patches of pericycle. Okay. Then in the center you will have pith. Okay. And then you will have secondary xylem, and then in that secondary xylem you will have included phloem. So we will draw secondary xylem here. So in this way, this is the structure, how it will be observed in a transverse section, and this will be formed by abnormality in the cambium behavior. Okay, let's label it. This will be epidermis. Okay, then you will have cortex. Hypodermis may also be present, and if the stem is slightly older, then periderm formation could also be there. Then you will have endodermis, pericycle. Okay, this will be secondary phloem. Then this will be secondary xylem, and then this will be included phloem. Okay. Some phloem you will also see in the pith region also. Okay. So actually leptademia is bicollateral. Okay. So bicollateral means that we have already discussed that you have phloem on the lower side also and on the upper side also. So in this case, you will see some phloem patches on the lower side also. So that phloem will be formed by these phloem which is present on the lower side. So because of that bicollateral bundles, the upper region will form the normal vascular tissue, but in that you will find included phloem, and some phloem patches will also be there in the pith region. Okay. Then in Salvadora stem also you will find included phloem. So in this case also again we can have the outline diagram. Again you can have epidermis, then hypodermis and cortex and that all. Okay, so in this case also you will see presence of included phloem. So let's draw it completely. Okay, so this is included phloem. And this is the secondary xylem, and this is the secondary phloem, and outer regions as we have discussed several times. Okay, here you get it appears that some sort of gaps are there between the secondary regions of vascular tissue, but this is not actually gap, these are medullary rays. Okay, sometimes you will see that the vascular bundles or the secondary vascular tissues are not in a continuous band and they are interrupted, as we will discuss later also one example in which you will see multi-seriate medullary rays. So sometimes there is an interruption and the vascular bundles are appearing as they are separate entities because of the presence of medullary rays. So wide medullary rays are there. Okay, so because of that so many gaps will arise. So in this also, this is the cellular diagram. You can see here that this included phloem is arising in this, and this is all because of change in cambial activity at certain point of its growth. At certain other locations also it could be formed. Okay, here also you can see secondary phloem, then some stone cells. These are the sclerified, basically sclerenchyma only. Okay, then endodermis, cortex is having chloroplast, okay, then hypodermis, epidermis and cuticle. Now coming to one very important example of which is having the presence of included phloem is strychnose stem. Okay, in strychnose stem also you will see presence of two types of phloem. One is the interxylary phloem and the other is the intraxylary phloem. See, when you will cut the section of this strychnose stem, you will see definitely in the center you will see pith, okay, and then in this secondary xylem region you will see presence of lot of included phloem, and along with that in the pith regions also you will see some phloem which are known as intraxylary phloem, okay. So the outer side will be normal secondary phloem. So this is the normal TS of stem of strychnose. Okay. So here also you can see that presence of included phloem is there, which is known as interxylary phloem, and in this you will see intraxylary . Okay. Now the formation is of this particular included phloem. First of all, let's try to see this picture. See, this is also a section of stem which I have prepared, where you can see presence of a lot of big patches of phloem in the pith region. You can see here that just at the boundary of pith. See, this is pith. You can see the pith is also colored, because that also I have told you that when the stem gets older, it also get somewhat lignified. Okay, those parenchymatous cells may also undergo sclerification and lignification. So that's why they are now appearing as colored cell, but these are actually pith cells only. So in the boundary of those pith cells, you can see presence of this intraxylary phloem, and this just above the pith region you can see this is the secondary xylem, and you can easily make out that lot of vessels are there. Here you don't find any conjunctive tissue, because you see that vessels are continuous here in this entire band. Above it, patches of interxylary phloem is started to be formed, and depending on the type of species also, the amount of that included phloem can vary. It could be more in some species of strychnose, it could be less. Like in some species, more intraxylary patches you can observe, whereas in some more included phloem could also be there. But depending on the species, this variation could be there. So this is the included phloem. Above this, you can see the normal phloem. Okay, they have started for giving rise to this included phloem. Now, how this included phloem and intraxillary phloem is formed? See, actually in this case also you can say that it is formed by R cambium method, but in this case there is no accessory cambia now here also. Okay, in this there is a normal secondary, there is a normal cambium ring. Okay, it is giving rise to xylem, it is giving rise to phloem, normal cambium. So, what happens in this? Suppose this is the cambium ring. Okay, in this also at certain points it becomes inactive. Okay, this cambia becomes inactive. Now, this inactive cambia, so now if it is becoming inactive, it will not produce xylem and phloem on both the sides. So, what will happen after some time? You will see this type of, because xylem is not increasing on the lower side. So, this type of depressions in these particular areas you will see, and some sort of phloem will be coming out in the xylem layer. So, what will happen after some time, and this phloem is definitely there, but in this case sorry, it's not this way. Okay. So, what will happen here? Xylem is definitely there. Above this phloem, definitely it's not growing in this way, because this cambia is not adding tissues. Okay, so this phloem will not grow in this way. This will also not grow in this way, although we are not showing the adding of xylem because this is on the downward side. After some time, what will happen? A cambia at this particular point in the phloem region or in the parenchyma, which is slightly above phloem, maybe somewhat this position or this position, they will start becoming active. Okay, they will start becoming active, and then they will, now this cambia will become active and they will join the original cambia. They will join the original cambia. Now, this is actually phloem. So, now and they will start behaving normally and they will cut xylem and they will cut phloem. So, in this way phloem, this phloem patch will come inside xylem. Okay, so you can say that this is somewhat like R cambia, that some sort of cambia on just above that phloem region. Okay, they will start becoming active and they will join the original cambia, and in that way that phloem patch will become embedded in the xylem. Talking about the intraxillary phloem. Okay, so in this case as in leptademia, we have seen that some phloem was also there in the pith region, but that was because of that bicollateral bundle. However, in the pith region also some cambia will become active. Okay, some cambia will become active in the pith region, means some, you can say some accessory cambia will be formed, the pith cells will become active and they will cut xylem and phloem, but in opposite direction. Okay, they will cut phloem on the inner side. Okay, normally phloem is cut on the outer side, but phloem will be cut, phloem will be cut on the inner side and xylem on the out. So, this phloem patch will come out. However, xylem will be mixed with the primary xylem, so it is not easy to differentiate, but phloem patches will be seen. Similarly, and in trachoma also you will see the presence of this intraxillary phloem. Okay, in this you don't find the presence of included phloem, however intraxillary phloem is there. So, it is also, you can see that because some cambia, internal cambia is arising in the pith region, it is cutting both xylem and phloem, but in the opposite direction. Okay, it is making phloem on the inner side. This is the phloem and this is the xylem. So, in this way you can see a patches of phloem in the pith region. See, this is just the cellular diagram which tells you that how this phloem, internal phloem, you can call it as internal phloem also, it is coming out in the lower side, it is just above pith, okay, and cambia is there and xylem is there, which will be mixed with the primary xylem, but maybe sometimes an extra, because it is cutting on two sides, so you can say that some extra xylem you could also see in the pith region, but definitely you will see the phloem. Okay, so this is all about our discussion that how secondary structures are arising in plant, and we have talked about dicot stems. Okay, so till now whatever we are talking was about dicot stem. In the next lecture we will try to discuss some anomaly which is arising in monocot stem as well as in some roots also we will try to discuss. So for today, thank you.